good evening and welcome to tonight's salon. I'm Adam Smith and I'll be guiding our conversation this evening, which is on working with brain tissue in dementia research. So brain tissue provides us with a unique window into biological changes behind memory, identity and decline. And unlike scans or models, it lets us see what really happens at a cellular level. But what comes practically, ethically and emotionally with that are challenges. And of course, more importantly than all of that is the practical ones. And yeah, I've completely messed up my introduction. So there we go. That's the first time in 60 salon sessions I completely lost my train of thought. But anyway, most importantly, joining us are two brilliant researchers this evening. We have the incredible Dr. Lauren Walker, an Alzheimer's Research UK fellow, I believe, at Newcastle University. Lauren's work focused on neuropathology of Lewy body dementia and Alzheimer's with a particular interest in co-pathologies. And alongside Lauren, we have the unstoppable Dr. Daniel Ruiz Gabarre, who is a research fellow in the UK Dementia Research Institute at the University of Edinburgh. He studies molecular mechanisms of small vessel disease and vascular dementias, whilst also being deeply engaged in equality and diversity in STEM. Some of you will remember Daniel from one of our very earliest, I think, is this your second time here now, Daniel? Daniel's not got his camera on, he's going to come back. I think this is your second or third time.
This is my second. I did the unconscious bias one.
Unconscious bias, that's it. So if you want to see, you can go back and watch Daniel's session on unconscious. So we, as ever with the salon, you'll be aware that we start with each of our speakers give a 10-15 minute talk, and then we have a chat and we ask them questions, and of course we also invite you to put your own questions forward, or if you really like, and nobody's taken me up on this offer yet, you're welcome to join us on screen as well if you want to put your question in person to Lauren or Daniel. That's enough from me, I think. We'll get started now, and Lauren has kindly agreed to go first. So, Lauren, I'll invite you to share your screen and I'll stick around while we check that that definitely works.
Are we all good?
We are. Thank you very much. Lauren, over to you.
Excellent. So, yeah, firstly, I just want to say thank you for having me. I think this is, it's the first time that I've joined this kind of session. So I think it's a very interesting concept. As I say, I do have a set of slides, but they're more kind of talking points. So happy to answer any questions at the end. So when I was preparing this talk, as we all do nowadays, I consulted ChatGPT and asked what it thought that someone that worked in neuropathology looked like. And just for kind of background reference, I work in neuropathology, so specifically the histology part of kind of brain tissue. And this is what it gave me. And unsurprisingly, I'm not very happy about this image for a lot of reasons. Mainly, I don't look like that. And I imagine a lot of people that work in dementia research don't look like that. One thing that is true about it, it is, by the looks of it, and it's in a dark room that doesn't really have any windows. And that is actually true for some of our labs. So that, I think, is the only part that's accurate. So whilst obviously this scientist does have a microscope and is clearly looking at some brain images, obviously this image is very outdated and neuropathology doesn't really look much like that anymore. It can do, but for the most part, and for future research, it doesn't really represent what we're trying to do today. So this is what a lot of people imagine that dementia research in terms of neuropathology looks like. It's very outdated. It can be used for diagnosing neuropathology and diagnosing dementia. However, it can play a role in multiple parts of dementia research and not just the confirmation tag on at the end of a study to go, well, hang on, this person had Alzheimer's disease, this person had dementia with Lewy bodies. So what I'm going to do is kind of go through some slides that kind of explain why I think this has developed from these kind of earlier stages. And to do that, I'm going to use this very it probably is old now, kind of Back to the Future kind of program, which I think ties in really nicely with the fact that Michael J. Fox has obviously publicly disclosed his Parkinson's disease diagnosis and has done for a long time. And I think neuropathology, it obviously does have a big past, but I think the present research that's going on is very interesting, and I think the future for neuropathology is very bright. I say that in a completely biased way, of course. So obviously neuropathology in the past has had some pretty major discoveries, from going back to the 1900s when Alzheimer identified things like plaques and tangles. Frederick Levy identified things like Lewy bodies, and then that moved through time when we were able to get more protein biochemistry advances, where antibodies were able to detect the proteins within these aggregates. And then we move on to the 90s, where we identified APP mutations, and this is where the amyloid cascade hypothesis came into play, and that's been a very central hypothesis throughout dementia research. Obviously, I think the biggest part of neuropathology in terms of kind of staging and diagnostics came in the 1990s and the 2000s, when there was a lot of kind of criteria that were established. So things like BRAC for tau pathology, tau for A-beta, and things like the multiple alpha-synuclein staging criteria that we have. And they've been excellent in allowing us to identify and diagnose the disease, shows us how far the disease has progressed throughout the brain. And obviously, this is very different to using animal models. So animal models obviously just give us a general idea of the pathology. It doesn't show us the human condition. So although these are historical findings, they still play a big role in dementia research today. And also in kind of 2010, mixed pathologies were recognized. And I've mentioned this here because this is my particular area of expertise. We've known for a long time that pathologies don't, or dementias don't exist as a single disease entity, and mixed pathologies are more common than single diseases. So for example, 50% of DLB cases will have a huge amount of AD-related pathology, and this then has implications clinically and pathologically for the patient. So neuropathology has played a big role in this kind of research area. And I'd just like to spend a minute or so highlighting some of the present dementia research using pathology. And I've got a couple of slides here that is based on my current work from my AI UK fellowship. So I investigated cerebral amyloid angiopathy, which is where amyloid beta deposits in the blood vessels. And using neuropathology and kind of quantitative neuropathology, we're able to start to classify dementia subtypes. So it's not just a case of this person has Alzheimer's disease, this person has dementia with Lewy bodies, this person has Parkinson's disease dementia, this person has Parkinson's disease. When we look at specific pathologies, kind of in a holistic view, we can see that in terms of dementia with Lewy bodies, it has a more similar profile. Some of the cases have a more similar profile to Alzheimer's disease compared to the other synucleinopathies. And this is very important when we start to think about clinical trials. So are we able to classify these patients, not just on the disease that they have, but on the mixed pathologies that they have as well? And this plays a big role in the stratification for clinical trials. It's also a big problem when we start to think about when disease-modifying therapies are available, and we need to be able to identify which patients will benefit from those. And that's not just basically this person has Alzheimer's disease, this person has dementia with Lewy bodies. The subtypes within this are a lot more discrete. So I think this is a role of pathology that can help us define these diseases a lot more. And also things like pathology can play a big role in blood biomarker research. So at the minute, there's 2 big clinical trials being carried out on blood biomarker research. And this is mainly looking at, I think it's the kind of tau biomarkers and A-beta. And these kind of biomarkers will tell you what levels there are in the blood. Obviously, we know that A-beta is reduced in Alzheimer's disease, and tau is increased in Alzheimer's disease in blood samples, in plasma samples. However, what we don't know is what this looks like in the actual brain tissue. So the markers in the blood are a proxy for what's happening in the brain. And I still think we need to consider this when we're designing these challenges, when we're designing these clinical trials. So this is just some data that unpublished data that I have so far. So I looked at p-tau181, which is one of the candidate tau biomarkers, and measured the percentage of immunopositivity in an Alzheimer's cohort, a DLB cohort, a mixed dementia cohort, and a control. And not surprisingly, the Alzheimer's disease had more of this tau pathology in the temporal cortex compared to the other diseases. However, the AD-DLB cohort, so the mixed pathology cohort, there was no difference between that and Alzheimer's disease, no difference between that and DLB, and no difference to controls. So for people that have mixed dementia, they can have a huge spread of this amount of pathology in the brain. And I think although blood samples will give us a lot of information, for a lot of these patients, it just won't be accurate enough. So I think future studies that we need to include neuropathology need to include the brain tissue as well as the blood samples. Unfortunately, we don't really have those cohorts at the minute. I know we take a lot of blood samples, and hopefully we won't get the brain tissue for these patients for a long time in terms of the patients, but I still think it will be important to include this donated tissue in the future. So moving forward, as Doc Brown used the DeLorean in Back to the Future, we as scientists also need to use new technology to help us crack the puzzle that is dementia. And I think, yes, there's a lot of new sexy science in omics, but in terms of neuropathology, there's a lot of technology that we can harness to increase our power as well. One of these is machine learning and AI, and I think this has a major role to play in the future of neuropathology. You've probably seen a lot of this image before. Is it a chihuahua or is it a muffin? And we're teaming with a lot of data scientists to be able to make machine learning and deep learning models to be able to do a lot of the legwork ourselves, take away a lot of the legwork that we do, and hopefully create models that will allow us to be a lot more high throughput, because neuropathology can be very time consuming. Obviously, there's a lot of problems surrounding AI, and I don't think it will ever take the place of researchers, thankfully, so I don't think it'll put me out of a job anytime soon. But I do think it will be a useful tool, and people are starting to develop AI programs and machine learning that allows us to identify pathologies. It starts to recognize pathologies such as tau, A, beta, and we're able to go through a whole host of tissue, very high throughput, and allow us to produce a lot more work, a lot more reliable, and a lot faster as well. And I think something that is worth saying as well, and I saw this diagram a few weeks ago, and it was a term that I'd not come across before, and it's called pathomics. Obviously, we have a lot of sexy science in the omics field, and I think it's important to remember that pathology and pathomics does have an important role to play in this, in the fact that, yes, people that work in pathology do look at specific protein aggregates, so things like tau, A beta, alpha-synuclein, but also in terms of pathology, we can look at neuroinflammation, we look at synaptic loss, we can look at autophagy deficits, and human tissue for this is an excellent resource because it actually represents lots of different time points in the brain. So I think what's important is that we marry up all of these technologies, and it was another term that I read somewhere, transformative neuropathology, kind of marry up things like radiomics, spatial proteomics, spatial transcriptomics, as well as other methods of looking at tissue that will allow us to get a better understanding from all aspects of the disease pathogenesis. And just a quick plug here for the Brains for Dementia Research Programme. This is an initiative in the UK that hosts, I think it's over 700 postmortem brains that's been collected so far. And this is nationwide. And in addition to well-characterized postmortem tissue, it has blood samples, it has DNA, RNA, and CSF samples. Stem cells are taken directly from the tissue and kind of modeled into iPSC lines. We have a lot of genomics data that Keely Brooks in Nottingham and Trent has done a lot of work on. And these tissue also had, the patients had a lot of cognitive assessments during life. So as well as actually using the human tissue, we can use multiple kind of strategies to look at the brain and what happens in the brain through multiple ways of looking at this and using different technologies to allow us to do this. And I think finally, I'd just like to talk about some of the challenges. I'm sure Danny's going to talk about some challenges as well. But just to cover a couple, just to finish off here. Obviously, brain tissue, I know this very well as a pathology researcher, costs money. Brain banks cost a lot to run. Obviously, there is the donation process. There is working with funeral directors, the storage of the tissue, the processing of the tissue. And this is now being passed on to researchers. So to use brain tissue in our research, I know that there's a cost recovery for this, which obviously is important to put in when you consider new research grants. For the last few years, I've needed to cost all of the brain tissue that I'm using into my research grants. And obviously this makes the research a lot more expensive. And also probably more importantly is the fact that the brain tissue that we get is taken from people. And we have a lot of problems sometimes recruiting people to the brain donation program. I think the first challenge is that not a lot of people realize, when you talk to people, they think that brain donation is tied up to organ donation. And so a lot of people are surprised when they ask about my work, and I tell them that it's a different register that they need to be signed up to. They're like, oh, well, I've got an organ card or an organ donation card. So brain donation is completely different to live organ donation. And it relies a lot on families. So obviously for a long time, we've needed consent for these patients. I think the HTA came in with that after the big scandal in Alderhay in 2004, which obviously rightly so. We need to consent the patient when they have capacity. And that's another problem as well. We need to make sure that the patient has capacity themselves if they've already been given a diagnosis of dementia, and to be able to consent to the donation. And particularly in the UK, which is different to other parts of the world, once the patient dies, we have to then get another lot of consent from the family. So we rely on the family to allow us to take the brain tissue. So we need them to contact the brain bank and sign another consent form so we can make the arrangements for the donation to happen. And obviously this is a very distressing time for families. They've just lost a loved one. So it is very difficult, and you need to kind of have compassion to be able to deal with this in a very respectful way, and understand that, yes, it's an incredible gift that we get from these patients. And we couldn't do our work without it, but they still are obviously going through a very difficult time, and they've lost their loved one. So I think it's just something that we need to keep in mind as well, that it is a difficult time, and we need to make sure that everyone is comfortable with the process as it's happening. So brain donation can be very difficult and very kind of emotive subject for a lot of people. So I think the lesson so far, brain tissue and particularly pathology is important in dementia research. It enables us to utilize information from all stages of the disease, and it will allow us to understand them better and ultimately design better treatments for patients. And pathology can play a pivotal role, and not just a descriptive role in dementia research. It can help us to understand a lot of pathophysiology, help us to discover if treatments are working, and help us to design better blood biomarkers. So I think I just want to finish off with one quote which came from the film Back to the Future. And I think it's very apt for dementia researchers like ourselves who are traveling roads that are not very certain. So one final quote: roads, where are we going? We don't need roads. So thank you very much, and happy to chat further.
Thank you, Lauren. That was brilliant. I really enjoyed that. And you covered so many topics, and I've got so many questions written down now. The first one I'm going to pick on, and I think this has come up on a podcast before. So the brain tissue you work with could have come from, if it's come from the brain bank, that comes with some information, right? What kind of information does the brain tissue come with that comes from the brain bank?
So it comes with lots of different things. Obviously, we get a lot. Every brain that comes into the brain bank gets a full set of neuropath diagnosis. So it'll have all of the Braak stage and the Tal phases, all of the different pathologies. It'll be staged. A lot of these patients during life also have clinical assessments. So things like the neuropsychiatric inventory, we have data on, particularly this is important for DLB patients, have information on hallucinations, auditory and visual hallucinations, depression, apathy. We have movement disorder skills. So the UPDRS gets carried out on a lot of these patients as well. And in the MBTR, so Newcastle does general kind of some genetic profile, and we have things like ApoE on all of our cases that come in through Newcastle. Things like PA.
So for anybody who's watching this, probably who isn't, not necessarily those that are live with us today, because I know that most of you watching are probably researchers, but if you're watching this later on YouTube on one of our catch-ups, so this isn't like a road, you know, where people suddenly die in a accident of some kind and then there's a donation of corneas or lungs or whatever it is. This is more pre-planned, that people would have consciously decided to donate their brain while they were alive, and information starts to be gathered so that when you get the brain, you know more about that person while they were alive.
Yeah, definitely. A lot of these patients are signed up for 10, 15 years before they pass on. So we do get, our research nurses go out and do annual visits. So we get multiple scores for each person, so we can capture the disease progression over. It's a really great snapshot to kind of capture over time at each specific time point.
So that's people both who, I guess, before people who cognitively, I don't know if healthy, normal is the right word, but people who are unaware of having some form of cognitive problem, and then, or who may go on to get it after they've decided to donate, and people who then get a diagnosis and go, oh, I'm willing to donate my brain, and you start. So different levels of information and different people and different brains. Yeah,
the people who don't have any cognitive impairment are just as important as the patients that do, and they go through the system in the exact same way. They get the exact same cognitive exams, even if they're showing no cognitive impairment, and the tissue is processed in the exact same way. So it is comparable to the people that had a diagnosis of dementia.
And something that I remember learning from you, I think you and Tamrin Lashley, I think, from a previous podcast, you should go check that out. We've covered this before in a podcast, about those that were given a certain diagnosis based on their symptoms or on whatever you could collect during life, compared to when you can then actually look at their brain to see what the difference is that I remember being quite surprised by how much your opinion can change when you can actually look at the tissue.
Yeah, so obviously we have excellent doctors who are able to give a clinical diagnosis, but that can be, I've heard it described as really messy sometimes. So people are complicated. They're not just one size fits all. And yeah, sometimes, and obviously I'm not a neuropathologist myself, but I've sat in on meetings in the Newcastle Brain Tissue Resource where obviously the medics looked at the clinical notes, and neuropathologist has given the neuropathological diagnosis, and there's no agreement. The neuropathology diagnosis isn't what they were given as a clinical diagnosis. And I think this is why it's obviously important to, if we could biopsy the brain during life, that would help us a lot, but that's obviously not going to be possible.
I wrote this week on science fiction and how science fiction often like what we read in science fiction obviously becomes a reality for like decades down the line. I can see little robots that go into your brain and collect stuff. I could see that becoming a thing.
Maybe, you never know. But yeah, it's definitely not as cut and dry as I originally thought when I started. I was like, well, obviously this person's got Alzheimer's disease, or we're diagnosed with Alzheimer's disease. And specifically when you consider things like mixed pathologies, which is what I'm interested in, there isn't such thing as a pure case. Even control tissue has some level of Alzheimer's related pathology there. It's just not enough to warrant a clinical diagnosis. So I think
so. That's quite incredible to me that that very few of the people who are diagnosed with plain run-of-the-mill Alzheimer's during life, when you then look at the pathology, actually had plain just Alzheimer's. They almost certainly had other things as well. It's quite astonishing, I think.
The things with pathology
are difficulty in subtyping exactly what they've got, I guess.
Yeah, it'll be interesting moving forward, because I don't think in the future we'll have plain Alzheimer's disease, plain dementia with Lewy bodies, plain Parkinson's disease. Ideally, for personalized medicine and to be able to treat patients, will be kind of my view, and it might not be shared by everyone, is that we'll need to describe these patients based on what's actually in the brain. So they might have TDP-43, they might have alpha-synuclein, they might have some RTAG, they might have some agrophilic grains, and to be able to identify, they might have something else that we've not even found yet. I mean, that's a kind of a negative about neuropathology, is it's very targeted. We know what we're looking for, where we have antibodies, we have these tools, and we're looking for a specific thing. And there might be something that we've just not come across yet.
Cancers are the same though, right? I mean, that's why you don't just say, oh, this person's got brain cancer. They actually have to look at what kind of cancer it is, because depending on the kind will then inform the treatment. Does that mean that we've got, sorry, Daniel, I'm keeping you waiting because I'm stuck in a track now, that the treatments we're currently looking at, things like anti-amyloid therapies, I'm guessing that's not necessarily a bad thing though, because is a common factor in these that they all do have amyloid. And so taking the amyloid, oh, you're going to say no now.
No, fully.
I mean, generally there's an agreement that amyloid doesn't belong there, right? And so an anti-amyloid therapy that takes away the amyloid is not necessarily a bad thing. However, because that might not be the only thing that's going on, do anti-amyloid therapies that I know this might not be your thing, do they help with those other things you've mentioned, like TDP-43 or those other bits that might be contributing?
Well, we don't know. The answer is, we know these pathologies or these proteins definitely interact. So we know tau and A-beta interact, and there are definitely trials that have been, or there's been calls for trials to test anti-amyloid drugs in Lewy body dementia. That's definitely going on in the States now, and I don't think that's a bad thing. Yes, they have a lot of risk associated with them, and we need to identify which patients will benefit. But patients that have DLB, that have amyloid pathology, decline a lot faster. So in an era where we don't have any disease-modifying therapies for Lewy body dementia, could their disease slow down if we give them an anti-amyloid? I think potentially. And there's some evidence to show that the amyloid that's targeted by donanemab, it's a species called pyroglutamylated amyloid beta. I'm going to plug one of my old publications now here. Neuropathologically, we found, it was a study we published in 2015, we found that that's associated with tau pathology. So if we reduce the amount of A-beta, could this have an effect on tau pathology? Could it lower tau pathology or stop more tangles forming? There's a complicated relationship between these proteins, all of these proteins, I think, that we haven't cracked yet. So it could potentially have an effect on other proteins. We don't know that answer yet.
So all of this, of course, me going down a slight rabbit hole there and getting a little bit more into the detail, highlights the importance that you wouldn't know any of this if you couldn't actually look at the brain tissue, particularly from those who very generously given up their brains to you through the Brain Bank and Brains for Dementia Research and other initiatives. Thank you very much, Lauren. We'll come back because I've got more questions, but let's hear from Daniel now. Thank you very much, Daniel, for being so patient. I know you're not using slides, so I'll just put the spotlight on you and let you take over.
Perfect, thank you, and no worries. Of course, like it was an interesting conversation, so I was happy to wait and enjoy it. Thank you for inviting me again. I am delighted to be here. This is like a really calm and safe space for this kind of discussion, and I really enjoy the opportunity. So for those of you who don't know me, my name is Dani. I use he, him pronouns, and I'm a Spanish neuroscientist now living in the UK. I moved to Edinburgh 2 years ago now to start my postdoc. I did my PhD back in Madrid, where I worked with Alzheimer's disease. There I did work with human tissue, and a lot of the things that Lauren was talking about, of course, resonate a lot with my research there. But since I moved here on my postdoc, I have started doing more research on small vessel disease, which is the main cause for vascular dementia. So it's related still to our topic, but a bit apart from Alzheimer's disease. And in this research, I use mainly preclinical models. More specifically, I use a knockout model for the protein ATP11B, and that's a rat model. So I work mainly with animals. So that's still working with brain tissue. I still do a lot of brain work, but it's mainly rat brains in my case. So I had a little chat with Lauren the other day to see what we were both going to talk about, what kind of ideas did we have. And yeah, just to try and maximize the diversity of point of views that all of you guys get here, or like will get if you see this on video later on. I am going to focus a bit more on that part of working with brain tissue instead. So I'm going to highlight a bit of what kind of things do we do with animal brains, and what are the limitations as well, as we discussed, and how do we bridge that gap in between me working with animals and how we translate that to the humans. Because at the end of the day, we do research to understand the mechanisms that happen in humans, both in health and disease. So one thing that I guess is the main point I would like to make is that even though working with animals is of course not as sensitive and delicate as working with humans, it still has, of course, a lot of ethical implications. It deserves the same degree of respect as working with human tissue. And I love that both Adam and Lauren mentioned the emotional implications and the need for compassion, because I think that applies throughout working with animals as well. Of course, the type of ethical implications that we have are different. So when we work with animal models, we don't really need anonymity, or we don't really need informed consent from them. But we do need to maintain animal welfare. Do we need to have a specific justification of why we use animals for this kind of research? And we need to ensure that everything we do is as safe as possible for the animal and as respectful with their lives as well. There's these principles of working with animals, which is general, not only for working with brain tissue, but just like working with animal models, which are the 3 R's. Those are R's for replacement, reduction, and refinement. So the idea is that we should replace animal models with something else if we have the option. So if there's like any in silico approaches, like computational methods to model the same disease or the same aspect of disease that we want to study, we should try to prioritize that. If we can refine any of the points of our work with animals, we should be doing that, so that it's not as harmful or not as painful or uncomfortable for the animals. And we should be try and reduce the numbers. So that's for that kind of thing is very important. Doing a thorough experimental design, having the statistics in mind before starting our experiments, and so on. And I know that like this might sound like a bit more of a boring part of working with animals or working with brain tissue in this case, but I think it's really, really important that I highlighted that before talking a bit about the work we do. So about that, like the fun part of working with animals, the good part of working with animals in this sense is it allows us to do a lot of things that we cannot really do when we work with brain tissue in humans, or when we work with humans in general. So Lauren was mentioning that we cannot like stop and like take a brain biopsy from a human at any given point. In animals, it's not common practice either to just like take a brain biopsy, but like if you do experimental design and you where you want to research what happens in the midpoint of the progression of a disease, you can just design your experiment for that. So you would get an earlier points on the development of disease if you work with animals, for instance, or you can choose what and when do you want to study. So it's a bit more flexible in that regard and offers a lot of insights that then we can translate to the kind of research that we do in humans, and allows us, of course, to better understand those mechanisms of disease in humans, hopefully with, yes, these ideas of finding new targets or exploring those mechanisms. I think one of the things that is very interesting there is, again, what Lauren was mentioning as sexy sciences, so all these omics. It's easier to do just, if we think about volumes, for instance, like the brain of a rat is something like this. So if I want to do like a whole brain proteomics or transcriptomics, so that would be all of the proteins in that brain, or all of the RNA molecules in that brain at a given point, it's easier to do. It's just like more manageable with animal models, with rats in this case, than it is with humans. And that's a bit what I'm doing right now. Like my research right now is focusing on building a transcriptomic and proteomic profile of this rat model that I mentioned before, which is a model for small vessel disease. So it would help us understand what kind of things are, what kind of mechanisms are causing vascular dementia, or like in this case, small vessel disease as a cause of vascular dementia. And then we can just try and translate those same pathways to the humans, because even though animal models and humans, we are really really different. A lot of the mechanisms that we share for this kind of diseases can be pinpointed in the animals and then translated to the humans. One of the most interesting things I think we're doing currently in my lab in this regard is we are sort of doing what we've come to call reverse translation. So it'll be the other way around to what I was just describing. There's the group of Joanna Wardlow in the UKDRI have been doing these clinical trials with humans. They're called the Lucky trials. And they have explored the use of two different medications for, well, they're like endothelial cell activators, but they're trying to apply them for small vessel disease. So they use ISMN and cilostazol, either alone or in combination. And they have seen that people with small vessel disease and people with stroke improve. So we don't really know a lot about the mechanisms about how those drugs are working in those patients. So what we are trying to do now is we took those same drugs, dosing regimes, and we have like put them in rats and have tried to emulate that clinical trial on the animals. So we're doing this reverse translation to do like preclinical animal trials with these same drugs. And this is, again, something that we can highlight as one of the advantages of working with animals, that we can stop. So we're doing younger animals and like older animals, and we are like comparing the results of these drugs in both of them. So that way we can understand mechanisms, like earlier mechanisms and later mechanisms on small vessel disease, which would be invaluable in the future to try and prevent disease instead of trying to treat diseases. Then also I wanted to mention some of the challenges of working with animals. So this is all of the positive things we can do with them, and that is fantastic. I really enjoy my work, but again, it does have challenges. So apart from the very obvious ethical and emotional tax that this, like working with animals, can have on you. Like at the end of the day, for instance, in this trial, we are treating the same rats for about 7 months, and then we need to sacrifice them to get their brain tissue. So that's emotionally taxing in that sense. But apart from that, some of the challenges that we find are very similar to the ones that Lauren was mentioning to working with humans. So for instance, there's the main challenge in life and in research, I guess, which is money. So having animals have associated costs. So we do have to pay for keeping the animals in the animal house and keep like the space where we keep them, and for breeding them and growing the population and this kind of thing. There's other challenges which are also similar to humans but have like some particularities. So that's the tissue integrity and preservation. So in this regard, for instance, how do we measure how the tissue is preserved or how much it is degraded is one of the most important things to ensure reproducibility in research. So we can assure that whatever we do and whatever results we obtain will be valuable and will be more easily translatable to human disease. In this regard, just to, I guess, give a bit of a taster of like how is more pragmatically working with brain tissue in this aspect, I would say that one of the main measures of taking into account this tissue integrity preservation is post-mortem delay. So how much time since the death of the patient or the animal in this case has passed until you actually froze the tissue or use the tissue, depending on the kind of technique that you need to use. But I have found empirically with data from a lab that time postmortem is not as accurate in describing this. So we have come with a bit of a scale instead of how consistent, like what kind of consistency the tissue has, as a proxy of how thaw it is while you work with it, or how close to be frozen it is. And that has been more reproducible in between brains than just plain time, because there's other factors, not only time, but like other factors in how it was transported, or how much time it spent in ice with respect to in fresh buffer, or this kind of thing. Finally, on this kind of challenges, I would say there's a big limitation of working with animals that, of course, they do not fully represent. There's no single animal model that fully represent the equivalent disease in humans. We do our best and we get as close as we can, but yeah, it's never the same. There's also the problem of like we do have a reduced genetic pool when we have animal models. So you have the animal that you create, and then you breed it with other animals in the same litters or in adjacent litters, and that is still not representative of the kind of diversity that humans have. So even though like we try and get things as reproducible as possible and like as translational as possible, we do get a lot of times where things that are successful in preclinical trials or in preclinical efforts then are not successful when we move on to humans. And that, sadly, for now, is just the way of research. And well, I'm kind of glad, because that means I do have a job. So some of the things that I would like to pinpoint the most in here as sources of diversity are some of the ones that I chatted with Lauren about are sex differences, but also ethnic or racial differences, and regional specificity within the brain as well. So there's a lot of these things are factors that we can, to some extent, control in the animals, and we are failing to capture in humans, or in the translation to human disease. I would say sex is maybe easier to address as animal models than in humans, because it's a less diverse experience like that, at least from our point of view. I cannot ask the rats about that. And it's easier for me when I plan experiments to have the same number of males and females, for instance, in our litters or in our experiments, than it is to when you try and capture or try and recruit patients to absolutely guarantee that you have a 50-50. But again, you've got this problem, for instance, with when we come to like racial differences or like ethnic differences, you've got this problem with animals where if the gene pool is like really reduced, you are not really capturing any of that diversity. And there's a few studies regarding this, that not all ethnic or racial diversities experience disease, and in this case, dementia, in the same way. particularly on mixed pathology, as Lauren was talking about. African Americans and Caucasians do not show the same kind of proportions of, say, AD with respect to Lewy body dementia in between these groups, for instance. With all of these, I guess the conclusion to wrap it up is that none of these challenges are specific to working with animals, and a lot of them are certainly not specific to working with brain tissue. But I hope that sort of gives you a taster. And I did not have any quote prepared to finish, but I got jealous of Lauren having one. So I can go to my go-to. So in the words of Taylor Swift, I guess the thing we should remember when we do work with brain tissue, or with animals in this case, is never be so clever you forget to be kind.
Daniel, that was very good.
Thank you.
And an important message at the end there, which I'm going to pick up on, actually. You mentioned, so we didn't really cover this in Lauren's talk, it was about diversity. I mean, we know that the people who volunteer to participate in clinical trials don't really represent the population of people that develop dementia. I'm going to guess that it's the same for people who donate brain tissue. What's the diversity of brain tissue like?
Yeah, we were talking about that indeed. Also, we are very biased about where are we doing the research. So we're both based in the UK, like that's where this platform is from. So the kind of access to brain tissue and to brain banks, and even to the concept of brain donation that we have here, has nothing to do with the kind of access that is in other parts of the world. So just by that and by the kind of populations that we have in westernized parts of the world, most of the research we're doing is being done, at least in, as I said, in this part of the world, in Caucasian brains. Most of the patients that come and volunteer for this come from usually more socioeconomic, provided backgrounds.
Yeah,
it's again the same.
So, I know the UK brain banks in brain tissue. I remember watching a slide from the Bristol one recently, which we've got some talks on our YouTube channel, showing this great map where all the brain tissue literally gets sent all across the world from UK brain banks. Do we have access to brains from other parts of the world as well? Where does your brain tissue? Where can you access brain tissue from other than the UK?
Mm-hmm. I've not been working with brain banks since I moved to the UK, so I'm unsure about regulations within the UK. So maybe Lauren can enlighten us a bit more on that. But when I did do my my PhD in Spain and we used brain tissue there, we did have access to some. So, for instance, within the EU, it was easier to access. But some brain banks from other parts of the world were just forbidden, kind of legally for us. It was a real pain to try and go through that.
Yeah, just culturally, I guess, sensitivity. Mm-hmm. Where do they come from, Laura? Where do your brain samples come from?
So the majority of mine come from the UK. However, we've had tissue from US brain banks, and they're slightly better at diversity, but not as much as you think. They still have a lot of problems trying to recruit from non-white populations. I mean, it is a big problem in the UK, as Danny already mentioned. Things like socioeconomic status is a big problem. And people who hear about brain donation, they're not your average person. They're the people who have been to university, the people who will have heard about research and research trials and things that they can get involved with. So that's unfortunately not available to everyone. I always thought, because we're particularly bad in Newcastle, I was like, well, maybe if I go to the London brain banks. And I remember talking at a meeting with Steve Gentleman, and we were talking to, I think it was Brittany Duga who was giving a talk, and she's from the US. And Steve Gentleman said that they still have the same problems in London. It's not as diverse as the population is in London. As I said, the US are slightly better, and we do have kind of different backgrounds in some of our tissue. So we're trying we're trying to get better at it. And funders are now asking for that as well. A lot of my recent applications is, yes, as well as sex differences, how is your study going to cater to a more worldwide population? And we're kind of trying to find different brain banks that will allow us to get a combination of brains that will make it more even.
Oh, that's great. I mean, it's great because that's really, it's only, I guess funders are going to push that, but also as well, it's building in the cost, isn't it? Because I'm guessing it's not cheap. If there was a brain bank in Cape Town or Nigeria or in Sao Paulo or wherever it is, getting brain tissue from there is possible, but I'm guessing also expensive.
Yeah, definitely. We've got a colleague who, Rufus Akinyemi, who used to work with us in Newcastle, and he's setting up a brain bank in Nigeria. So
Wonderful.
that will come, but obviously that's going to take time to collect the tissue.
Well, because the lead time is, as you say, so long. I mean, some people have to be signed up for 10, 20, 30 years to get it's such a long lead-in time to. And what realistically do you learn? I mean, you know, what is there to be learned from making sure our brains are more diverse than just using this tissue? What are we missing by not having that diversity?
Well,
Daniel.
I guess to begin with, just representativity. Like we cannot take this tiny bit of the population and assume it will, whatever we find there, it's going to be expansive or translational to everyone else. We've seen this happening in clinical trials. Like we do try some drugs in clinical trials, and then when we try them in the general public, sometimes like a lot of more adverse effects happen, or they don't work as well. There's a lot of bias going on there, even in like the way we do research on the first place, and then we try to impose those things onto the rest of the population. That has happened historically when clinical trials only recruited men, for instance, and then we try to give the same treatment to everyone, and then people who are not men were not having the same success rates, or were having other adverse effects that were not thought of during the clinical trials. So just the idea of, if we are doing research that we are really proud to say it's applicable, then actually making it applicable. That's what the diversity would be able to guarantee.
And that's everything, isn't it? Right? I mean, it's just like the food we eat, the air we breathe, the, all these things that we know affect our brains. The water that you drink, I don't know, I'm assuming that can eventually, if you drink so much of a certain type of water, it's going to get to your brain. I'm thinking heavy metals now and things. We had some toxicologists on the podcast a few weeks ago that were talking about differences. I've got so many questions, but we literally have only 5 minutes left. We're going to speed through some of these, if it's even possible. What conversations should researchers be having with families and donors to build trust?
I think for me, it's giving an honest representation of what we do. I think a lot of people have visions of glass jars in basements of dementia research, and we do a lot of tours at the Newcastle Brain Tissue Resource, and people are amazed that how everything is meticulously categorized and meticulously stored. And you tell me what sample you want, and I can locate exactly what shelf it is on a freezer or in a space, and just kind of show the care that we give to the tissue and that we're appreciative of that, rather than just treating it as a random bit of tissue with a random number.
No single piece goes to waste, right? I mean, you literally, like, there's no bit so that's just disregarded, that every single... Daniel, sorry, you look like you had something to add.
Yeah, I would say they're like maybe a conversation that I think we should be having with everyone that is considering brain donation. There is that brains from people who are not expressing, like, say, dementia, or who have like other type of pathology, or do not have any brain pathology at all, are also needed, are as necessary as the ones from people that do have dementia. So yeah, just try and make everyone involved, because like societally we are all trying to move forward with this.
I mean, I think what's come across from both of your talks is the real sensitivity and sense of privilege that I know you have from having the opportunity to work with this tissue. How does the emotional weight of that prey on you? I mean, is this, I know when we get into, we've had a session earlier this year talking about animal research and the how it feels to have to work with animals and that. But I mean, I know these people have obviously died already. I mean, is this something that you emotionally causes you problems, or are you disconnect? Is this just tissue are there emotional elements to it for researchers thinking about, or who might be working with brain tissue for the first time?
Yeah, I would say it's never emotion free, at least if you're doing it right. Because at the end of the day, yeah, like it does belong to a person. It did belong to a person, or again, even if you were with animals, it did belong to an animal. So I think that level of respect and that level of like emotional implication should always be there. And I think it's actually like an useful emotion. It helps us, at least it helps me to focus and realize I'm doing this for a purpose. I'm just not like here passing my time today doing cell culture.
And I guess the difference here is, I mean, you must feel to some extent a bit of a connection, because whilst I'm guessing you don't, maybe if you work in the brain bank, you wouldn't have known these people while they were alive. And I'm guessing they're anonymous, but you just have their medical history, or can you kind of build a picture of what this person was like from their notes? And
you do get a bit of a clinical background, so you find out, particularly if in a previous life I worked as a technician in the brain bank before I did my PhD, so I was a lot more involved with the kind of donation and collection side. But you do kind of get a bit of a background. If you ask for like specific clinical information, there's normally a kind of an abridged version of clinical notes, like this person was doing crosswords up to this point during life, or this person lived alone, or this person. So you do kind of get a little bit of a background and a bit of a sense of the person. And I think, as Danny said, you have to realize why you're doing it, and you always have to bring it back to you're doing it for a reason. And yeah, specifically when you see, I always found this when you get the whole brains, when we're in a dissection room and we get whole brains, it's like this is essentially it's a person. So I don't think you can ever really, or I've never been able to completely remove the emotion from it.
I'm afraid I've got so many more questions, but we've only got 2 minutes left. And in those 2 minutes, I'm going to give you a couple of stats, and we're going to go through our top 10 tips. Then I'll come to that. So apparently around 40% of dementia diagnosis show mixed pathology. According to some stats from the MRC. In the UK, there are 10 dedicated brain banks forming the MRC's Brain Banks network. I thought there were more than that, actually, but the stats say 10. And public surveys show that while over 65% of people would consider donating their brain, only a small fraction of those people actually do. So if you're watching this on YouTube, have a look on the internet. We'll put a link in the show notes for Brains for Dementia Research, or there's other initiatives as well, aren't there, which we'll put links to there. Please do consider doing this. You will be helping and making a big difference, and what you can give after life is just as useful as donating an organ for transplant into somebody who would be carry on living. So we'll finish on, this is where we test our two speakers. So we have trained an AI model purely on content from Dementia Researcher's website. So this is not being trained on the internet. Dementia Researcher website has over 3,000 pages of content. This is podcast transcripts, webinar transcripts, blogs, articles, job descriptions, funding calls, you name it. It's been trained on everything, all those 3,000 plus pages on our website. And one of the things I ask it to do every week is to give me 10 top tips on takeaways on the topic of this week's Salon webinar. And I think it's always good to see if our two guests have picked up on the same issues. You usually have, but let's see. So number one, work with brain banks early. Relationships and planning are essential for timely, well-accessed, well-characterized brain tissue. Don't wait until you've got funding before dealing with the brain banks. I don't think you both specifically mentioned that, but we weren't talking about practicalities here, were we, today? Number two, always check metadata. Age, comorbidities, medical history, post-mortem delay can radically change interpretation. Number 5, respect donor intent. Remember, every sample is from a person and a family who made a conscious choice to donate. I think you both picked up on that very well. Integrate neuropathology with clinical data. Pathology alone is rarely enough. Clinical notes give context. I think you both did that too. Number 5, embrace co-pathologies. They're the rule, not the exception, and must be factored in study design. Well done. I think you tick that as well. All of this has come from your previous podcast, I suspect, Lauren.
I think so.
Number 6, validate findings across cohorts. Over-interpreting single tissue collections. Avoid over-interpreting single tissue collections by comparing across brain banks. Number 7, standardize your protocols. Fixation, staining, and storage differences can create misleading artifacts. There you go. Number 8, acknowledge the emotional weight. Teams benefit from open discussion about the sensitivities and of handling donated tissue. Number 9, link tissue research to biomarker work. Without neuropathology, validation of imaging and fluid biomarkers is impossible. Lauren did that one. And engage the public directly. Explaining why brain donation matters increases awareness and builds future resource. I think you all both mentioned that too. Thank you so much. You've both been really generous. It's late in the evening in the UK, and both Daniel and Lauren have generously given up their evening to join us. We'll be back in the salon next Wednesday at 12 noon, where we're going to have Ian Harrison from UCL, who I think Lauren's been on a podcast with before for us. And we've also got Rosie Jackson from the University of Dundee. So we're going to be back to Scotland, where we're going to be talking about the top tips in starting your first research lab. And then we've just advertised the very first of our salon debates. So we're going to have a few more salon webinars. And on the 15th of October, we're going to have our first salon debate, where Professor Michael Coleman is going to be arguing in favor of this motion, and Lucia Katsuri from UCL will be arguing against it, which is the House believes that animal models have outlived their usefulness in dementia research. So that's what the two people, Lucia and Michael, are going to be debating. And that's on the 15th of October, where we'll hope you get to poll you before to see what you think at the start of the debate. We'll poll you afterwards to see if you agree afterwards, just like the debates you see during the elections. We're going to have that one on animal models in dementia research. So I hope you can join us on the 15th of October. Thank you once again to the incredible Dr. Lauren Walker and the amazing Dr. Daniel Gabarre. You've both been brilliant. Thank you so much. And we'll see you next week in the salon. Thank you.
Thank you. Bye.
Bye.
Bye.